Hello viewers, welcome back to my channel, and today's topic is why is gram staining important. Gram staining is a very important and basic technique in microbiology and pathology lab, and it is also very important to learn why it is important on the basis of the viva question, for viva question, and also for the practical knowledge of microbiology. So let's start it. First of all, gram staining is a differential staining. It differentiates the broad category of bacteria, that is gram positive and gram negative. We all know that bacteria are classified into the two main category, gram positive bacteria and gram negative bacteria, and for the classification, for this classification, gram staining is very important. Okay, in gram positive bacteria, thick peptidoglycan layer present, and due to the presence of thick peptidoglycan layer, it's retain the crystal violet color and appear in purple color. Okay, and some example of gram positive is staphylococcus, streptococcus and lactobacillus. And in gram negative bacteria, they do not retain the crystal violet because they have very thin peptidoglycan layer and also present of lipopolysaccharide layer. Okay, in gram negative, thin peptidoglycan layer present and lipopolysaccharide layer present. That's why they do not retain the crystal violet. They retain the counter stain, saffron color. Okay, that's why it's appear pink in color when we observe under the microscope. Next, why it is important? Why gram staining is important? Gram staining is important for quick identification, help to rapidly categorize bacteria, whether it's gram positive or gram negative. It help and guided for the appropriate antibiotic therapy, understanding the bacterial structure, and it is also important for clinical significance, for the laboratory assessments, and it is a very cost effective and simple because it is easy, rapidly inexpensive technique with great values. Okay, in short, gram staining gives a lot of information in just a few minutes and is the foundation of clinical microbiology. Okay, and here is the simple step of this technique. First of all, we use primary stain, after moderate iodine we use and decolorizer, and in last we use saffron. Okay, so this is all about